says, the first part of this says, complete the right side of the DNA molecule by matching the complementary bases to form complete rings on the latter. Note that on the left side, the 3 end should be exposed at the top and the 5 end at the bottom. Your completed model should look like a ladder with matched bases forming the rungs. The DNA molecule twists, however, your model cannot show this. Name the 2 molecules which alternate to form the portions or handrails of the DNA molecule. So remember, you have a sugar, a pentose sugar. You have, depending on whether you're making RNA or DNA. If it's a DNA, it's a 5-pentose sugar. It's oxygens. You have phosphate. You have nitrogen, and you have the bases. The bases are nitrogenous bases. So the handrails are made from sugar and phosphate. Sugar and phosphate. Name the specific molecule within the nucleotide to which its base is attached. Okay, they're attached with hydrogen bonds to each other. Okay, they're attached with hydrogens. What did you have to do to the nucleotide models on the right in order to match them up to the complementary nucleotides on the left? So you had to match the bases, and then you had to connect the 3 to the 5 and the 5 to the 3. We watched a video about this connection last couple of sessions ago. Is the order of bases exactly the same on each side of the DNA molecule? Well, they're the same bases always pair with each other, but the order is not the same depending on the type of protein that you're trying to make. It depends on, and you have to match the 5 ends to the 3 end. So, of the 4 bases, only specific ones can bind with each other. List the complementary pairs: thymine and adenine, okay, guanine and cytosine. If 4 guanine nucleotides appear in a DNA molecule, how many cytosine nucleotides should there be? Well, there should be 4 because each of those will pair with a guanine. So you got that. The following are the bases on one side of a DNA molecule. List the bases that would appear on the opposite side. So here you have thymine with adenine, adenine with thymine, guanine with cytosine, guanine with cytosine, cytosine with guanine, adenine with thymine. You would have uracil if you were making RNA. RNA is the single-stranded molecule. Okay. The DNA molecule you have constructed is only a short segment of the actual chromosome. An entire chromosome has thousands of base pairs rather than only 9. However, we can use your model to simulate the process that replicates DNA prior to cell division. So that's that activity. Then you have transcription. So the first step, and we watched a video about this too. The first step is translation. So you have, is this the right one? I think not. I think this is not the right one. Let me open up the screenshot because I don't think it's showing you the right. The right. Can you see the activity 3?
Yes, I can see him.
Okay. So the first step in the synthesis of a protein is to transcribe the genetic information from a DNA into an RNA molecule. And then it gives you the procedures of what you did in the lab. And here he has a note for you. In RNA, the base uracil takes the place of thymine, which DNA nucleotides are complementary to uracil nucleotides, okay? Remember they're replaced. Carefully remove your RNA segment. Okay, so now, okay, so in DNA, is ribose present? No, because DNA has deoxyribose. That's what the D stands for. Is deoxyribose present? Yes. Is phosphate present? Yes. Is adenine present? That's one of the nucleotides. Is thymine present? Yes. Is uracil present? No. Because he told you here in number 4, the base uracil takes the place of thymine in RNA. So uracil is present in RNA, but not in DNA. Guanine, yes. Cytosine, yes. But in RNA, it's not, because that's the one that gets replaced with uracil. Cytosine gets replaced with uracil. Is it double-stranded DNA? Deoxyribose, 2 oxygen and ribose. So double-stranded, no for RNA. Single-stranded, yes for RNA. Does it remain in the nucleus? DNA does, RNA travels outside. Does DNA leave the nucleus? No, but RNA leaves the nucleus. Okay. So that's part of those characteristics that he's probably going to test you on. Okay. Okay, let's look at the next screenshot here. Okay, now we're going to look at our next screenshot. Oh, and grab this out. And I think it's this one here. Yeah, it is. Okay, so this is activity 4, translation. The mRNA, remember that's the one that leaves the nucleus, moves to the ribosome. Do you remember what happens on the ribosomes? There's something. What does the ribosome do for you in your cells? What is the purpose of that?
I don't really remember.
Okay, so RNA travels outside the nucleus to the ribosome. The ribosome is where proteins are put together. So remember here that I'm going to put this in the chat. Ribosomes are sites of protein synthesis. Synthesis means putting things together, or assembly. In the cytoplasm, remember, not the nucleus of the cell. So the DNA stays in the nucleus, but the RNA travels from the nucleus to the ribosome, and that's where all of this is taking place. So this coded piece of protein is going out to the ribosome. It's carried by messenger RNA. Then it gets transcribed, it gets written, and then it gets translated. Okay, so what you've shown here is the first lab showed you the DNA molecule. The second part showed you the first process, which was transcription. Now it's translation. So it says the mRNA, that's messenger RNA, moves to the ribosome. It contains information in its sequence of bases, which are complementary to those of the DNA. At the ribosome, the messenger RNA controls the synthesis of the protein by ordering the sequence of amino acid building blocks. The amino acids are carried to the ribosome by tRNA molecules, transfer RNA molecules. You will now translate the messenger RNA molecule that you transcribed, forming a 3 amino acid protein. So which transfer RNA anticoding will bind to the mRNA codon of GUC? Here you have to use the complementary pairs. So guanine, which is the G, we need the complement for guanine. The complement for guanine would be, so here's the codon. So instead of just looking at one nucleotide, you're looking at a whole codon here. So this is guanine. Guanine we know pairs with cytosine. So cytosine here. Then uracil we don't have thymine, remember? So it pairs with adenine. And cytosine here would pair with guanine over here. How many bases of messenger RNA are needed to code for one amino acid? 3. That's where this 3 comes in. A protein molecule sequence contains the following amino acid sequence: leucine, LEU, tyrosine, TYR, serine, SDR, phenylalanine, PHE. To provide instructions to the ribosome, what would the sequences of bases be? So he had you look up the codons for leucine, tyrosine, serine, phenylalanine. So you had to look up the codon, and then you had to write the instruction sequence for it. UUG, UAC, and that would be 12 bases, because 3 for each, there are 4 proteins. A ribosome receives the following RNA message. What will be the sequence of amino acids when translated? Okay, so what they wanted you to write here was not the names. They wanted you to write the base sequences. So AAA would be UUU. CGA would be the base complement of C, which would be G. So this would be GCU. They wanted you to write the sequence of amino acids. And those would be the amino acids underneath it: lysine, arginine, glutamine, and valine. But you started to write it up here, which is not a bad idea. Good job. So now we're going to go to the next one. Let me get your next screenshot here. Okay, here's your tables. Okay. These are just the tables that you had this is your next, that's just the table of now we have, that was how you came up with the And then we have the next screenshot. Okay, so now, can you see number 5?
Yes, I can see it.
So now what we're doing here is you have the let me move it up so I can see the box. Okay, so we have for now. We have the DNA codon TCG. So we know A pairs with T, C pairs with G, so that's guanine, and G pairs with C, so that would be AGC. And then they want to know what the amino acid would be. So your codon they want to know what your amino acid is. So here, that's where you have to go back to your chart and look at your amino acid. So it's A, and then the second base. So here, so if you have AGC, the first base is A. The second base is G. So AG, you'd be here somewhere, okay? And then you need the third base is over here. So AGC would be serine. Serine is your amino acid. So when you get to this base, when you're doing this page, I think it's this one. Let me make sure. No, that's the wrong one. When you get to this page here, let me figure out which screenshot this is. So your first base, like I said, is A, then G, then C, so serine. So what they wanted you to do was, on this one, was you did the mRNA codon. So the amino acid that you would be coding for would be serine. So you would have put serine here. Because according to the chart, that's serine. The second one is GGG, so that would have been CCC. So then you would have gone back to your other page. If you go back to the other page, it's a CCC. So you've got the first base you have is C right here. Then the second base is C. And the third base is also C. So the second base is C. The first base is C. So C, 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 that would be proline right here. That would be proline. So this is where you have serine. So this is serine. This is proline. Now here you have A pairs with U, T pairs with A, and A pairs with U. So UAU, if I take this back now and I say, okay, and I go here, UAU, this is my other, so I have serine, I have proline. This now, the first one here is U, so I have to be up here. U, A, U. So the third base here, U, A, and the third base is U here. So this is going to be triazine. So my third base here would be triazine. So this is, now I have this. Now I have serine, proline, and I'm working on the next, hold on. So this becomes there it is. Now I have serine, proline, triazine, UUU comes from AAA. So UUU we did on the previous one. So if I go back and I share this one, the first base is U, The second base is U. So that puts me in this column. And the third base is also U. So I have the U here, that's phenylalanine. Phenylalanine. So here I have so this was serine, proline, triazine, and phenylalanine. So now I have this is hold on. This is serine, proline, triazine, phenylalanine, and CAA. Let's look at CAA. What is that? Well, CAA, here's my first base C, my second base is A, and my third base is A. So it's a stop codon. BA, A, my third base. This is a stop codon. So serine, proline, triazine, phenylalanine, and stop. So there's a total of 12. Okay. That was for this. That was for this page. Because he wants you to name the amino acids over here. So this was serine, proline, triazine, phenylalanine, and then the last one was a stop codon. Then we had activity 5, which is mutational. Talks about mutations, and it talks about hemoglobin and sickle cell disease. So let me get the next screen share from here. Get the next file. We only have 2 more. Okay, so this is talking about forensic analysis of RFLPs. Okay, so let's look at this here. Let's see. We've got, oh, second, there it is. Here's my screen share right here. Okay, so it says activity 7, forensic analysis of FLPs. The following information is for the photograph of the gel in figure 3.4. It contains DNA samples relative to a hypothetical crime scene. Okay. So you've got information on the side of this. You've got information right here. So there's a size reference. Let me use the pointer here. We have size reference. So we're going to reference this by the size of what we're seeing. We have our control strand of DNA, our other size reference, our victim strand, suspect number 1's DNA sample 2, and the size reference of what we're talking about. So lines 1, 2, and 3 contain controlled DNA and segments used for size reference. So here's 1, 2, 3. Okay. Line 4 is the victim's DNA. So victim, the DNA is here. Line 5 is from suspect 1. Suspect 1 is this lane here. Okay, so line 7 and 8, or sorry, line 6 is from the second suspect. So the one next to it, right here. Okay. Lines 7 and 8 are DNA samples collected at the crime scene. So here and here. Here, line, and here. And then this is your size reference again. So which suspect can be eliminated from this analysis? Well, if we look at the victim's DNA, which is right here, this is the victim's DNA, right? It has a match with this. And it has a match with this one, right? So which line is this? Well, this line is suspect 2. So this suspect can be eliminated. Okay, it says examine the gel and compare the pattern of the DNA fragments. Okay, and answer the questions below. So here, the DNA of suspect 2 matches. Okay, it doesn't match suspect 1. It matches the victim. It matches the victim, so you should change that. The DNA of sample, here's suspect 1. See, suspect 1, here's the victim, and here's suspect 1. The victim is this line here. It doesn't match suspect 1, but it matches suspect 2. So this should be the DNA of sample 2 matches the DNA of the victim. Other than a suspect's DNA, who else's DNA is left at the crime scene? The victim. Yeah, so it matches the victim's DNA. Okay. Okay, let's look at okay, here's another one. This is similar. This is using fingerprints, but to establish parenting. So it says, if I can find the Zoom link, we're looking at, there we go. Oh, my screen share is paused, that's why. Okay, so here we're doing fingerprints of, okay, there it is. Now we have every child gets half of his or her DNA from each parent. Therefore, comparing DNA fragment patterns from the mother and child will give you a partial match. The DNA bands in the child that are not present in the mother must have come from the father. While all of the bands present in each parent will not be in the child, bands that appear in the child's fingerprint must be found in either the mother's or father's. Observe the hypothetical Joe shown below and answer the questions that follow. So, okay, so this line is DNA from the mother. This line's DNA from the child. This line's DNA from the first man that's presumed to be the father, and this one is the second one. So if we look at the child, the mother's first band matches the child. The second band doesn't match anybody right here. The third band here matches DNA from dad number one. And the fourth band here matches the mom. So this match right here, how many of the mother's bands are found in the child? Well, there's one here. That's one. There's one band. This is a double band, so it's not this one, but it's this band here, one. Then one down here, that's two. And then one down here, that's three. So three bands in the child that are in the mother. And then there's two of the father's bands right here. These two match. And it says dad number one, and I agree with that. That's dad number one. Very good. And that was the end of that lab. That's as much as that was a lot to go over in one. That's in one lab.
And then I was wondering if you could maybe like find like some practice like problems on that.
You mean the matching band or the DNA replication stuff?
Yeah, I think we could do the matching today and then we could do the replication tomorrow.
Okay, so you wanna match, you wanna do something similar to the DNA match or the victim match?
Yes.
Okay, let me take